Hello, very good morning, students. I hope my voice is clear and I am audible. Okay. So today's topic is about the nucleic acid topology. So we are about to start with this nucleic acid. See, we have seen the important parts in the cell biology, like we have seen the transcription, translation, DNA replication. So right now we are going to see some of the basic parts that has been remained out of this topic. So in that part there is a point that is called a nucleic acid topology. See, in this entire lecture series, even if the points will be scattered, but you will find all of the points mentioned as they are in this UPSC syllabus. So we are going to have an lectures on each and every topic. Okay. So this nucleic acid topology is one of those topic. Okay. So yes, good morning, Guru Prashantam. Good morning to all. So in this nucleic acid topology, there is a thing that in many books or in notes you may find that the notes or material only regarding this DNA only. Okay, but just remember this nucleic acid is also a DNA, also an RNA also. So you will have to give a topology means a structure, shape, and its how its presence, how it present. That all just consider the covers under this topology. Okay, at least main this structure and the function and the shape of that nucleic acid. So we will see individually the topology of DNA also, topology of RNA also. So let's start with the first DNA. Okay, so in this DNA is nothing but what deoxyribonucleic acid. Okay, so let's see. See, in this DNA or RNA, there are certain nucleotides are there, and those nucleotides are categorized into that is the purines and the pyrimidines. Okay, so in purines, there are two nucleotides are there. They are called as adenine, and the second one is the guanine. I will change the color of the marker to white so that it will be easy for you. Okay. So it would be this either adenine and guanine. These are two purines. Okay. Then there are two pyrimids like this cytosine and thymine. Okay. But in case of RNA only, other three are same. This adenine, guanine, cytosine are same. But this thymine is replaced by uracil. Okay. This is the U in case of RNA. That is the only change. Otherwise, all other things are same. Uridine. Okay. So let's see now what are the other component of this DNA. This DNA has this one thing that is the nucleotides or the nucleotide base. The second, it is having this deoxyribose sugar. It is having what? Deoxyribose sugar. And the third structure it has in its component is that is phosphate group. There would be a phosphate group which is attaching this nucleotide base and this deoxyribose sugar. So this will be the what? Phosphate group. Okay. So these are the main three component of deoxyribonucleic acid or DNA. In case this is the deoxyribose sugar in case of DNA, in case of the RNA it would be the ribose sugar. Okay, so that is the case we can say a structure. Then we will see this in this topology only. We will also have to see this. Sir, can you confirm what we have covered so far? Are we covering syllabus of zoology optional for UPSC? Yes, it is Gautam Guru who is asking this question. See, this entire lecture series is on this UPSC zoology only, right? So we have covered many topics amongst it cell biology chapter. Okay, if you have seen the cell biology syllabus of the UPSC, you will find these topics enlisted in that cell biology syllabus of the UPSC only. There are 13 chapters in the UPSC zoology. Okay, out of that, cell biology is one. So, as I am saying, we are going to have a lecture on each and every point that is mentioned in the UPSC syllabus. Okay, and I have earlier announced in this group also because we have all the also a second paid batch also on this cell biology in which there are much detailed notes and you will also get a printed notes in that course. Okay, here I will only able to just mention all things in short. So just concepts most likely I try to explain here. Okay, so let's see this Watson and Francis Crick model of the DNA. See what's this Watson and Crick Francis model of DNA gave is that this DNA composed of this only. We have seen this phosphate group. Okay, this phosphate. this sugar. Now we will have to change the color of this. Okay, so this is the phosphate group. Okay, then it will have a sugar molecule attached to it and a nucleotide base. So after it, a nucleotide is getting formed. You can see this nucleotide is forming. This is the base nucleotide base pair. This is the phosphate sugar which is forming the backbone of the DNA. This is the phosphate group which is attaching these two adjacent nucleotides. Now you can see here. Here you can see these are the phosphate groups which are forming the backbone of the DNA. Okay, and these are the nucleotide bases attached to it. And one such unit forms a complementary bonding with the another unit. Now what does this another mean? See, this entire DNA is a what? A double helix, means what? It has two strands of DNA. Two strands. On each strands there would be presence of nucleotides like this adenine, guanine, cytosine, thymine. Okay. And another strands there would be a complementary base pairing would be there. What is mean by complementary base pairing? See, the complementary base pairing means guanine will always attach or always form a bond with this cytosine. This adenine will always form a bond with thymine. Okay. And guanine will always form a only triple bond with this cytosine. This adenine will always form a double bond with thymine. So this is what a complementary base pairing is. Okay. So have you understood? You will always find an adenine bind with the triple bond with thymine. You will double bond. Oh, sorry, with the thymine. You will always find a guanine attached with the triple bond with the cytosine. Okay. This is what the complementary base pairing is. So if here is the adenine, on this strand is there is an adenine, okay, and this strand. So what would be the next nucleotide on the another strand? It would be what? Thymine. Here same is the case with this guanine also. Okay. So in this way, this entire DNA strands are formed. The one strand is complementary to the another strand. Okay. So when this strand is present, but in this actual nucleus, they are present in a helical form. And why they are in a helical form? It is because of a conformational stability. Okay, this DNA is more stable in this helical structure than this. Okay. Now let's see what are the other things they are in this model of this Watson and Crick. So here I have explained you that how this adenine and guanine are attached with each other. Like you can see this is the one strand of DNA at this side. Okay, this is the complete one strand and this is the second strand. Now you can see adjacent nucleotides. This is the thymine and this is the guanine. So entire adjacent nucleotides are attached with this what? With the phosphate bond. But on the another stand, the same case, there is the same case. This is the nucleotide base. This is the phosphate sugar. This is the nucleotide base. This is the phosphate sugar, and these two nucleotide bases are attached with this phosphate link. Okay, but both of them have formed a bonding with the next strand. You can see this is the adenine, this is the thymine. As I have said, adenine will always attach with the double bond with the thymine. So that's why this is the double bond. And this is double bond will of what? This is the hydrogen bondings. Okay, will be there. Okay. Same case with the guanine and cytosine also. This is the three or the triple hydrogen bonding is there. Here you can see 1, 2, 3, and these are the two double bond 1 and 2. So this would be the case in this entire double helix model. And whatever these nucleotides I have shown you, these are those nucleotides: adenine, guanine, cytosine, thymine, uracil. This will be only in the case of what? RNA, and this will be present in the case of what? DNA. Okay. If you remember the structure, you can draw the structure. Otherwise, at least just mention the names. So in this Watson and Crick model only, there is another explanation, that is this model of DNA follows the Chargaff's rule. Now let's see what is the Chargaff's rule. See what is the Chargaff's rule is a very basic and easy terms. That is, Chargaff's rule is nothing but number of adenine will be equal to the number of thymine. And why it is so? I have already explained you that adenine will always attach itself with the thymine. This Chargaff's rule also states that this guanines, number of guanines will be similar to what? Cytosine. Why this would be the case? That because this guanine will only attach with the cytosine with a triple bond. So that is what the Chargaff's rule is, and this DNA obeys this Chargaff's rule. Then we will study what is the genetic code. See, genetic code is nothing but the sequence of DNA present on this DNA. Okay, number of, or you can say a sequence containing number of nucleotides in that DNA, and that nucleotide sequences or that DNA fragment will lead to the formation of a particular RNA. After that RNA is formed, it will ultimately form or ultimately translated into a kind of protein. So this entire terminology, this entire process is referred to as what? Gene expression. Now let's see in this, see we have seen a one model of DNA, but actually you can just explain this entire DNA model in as in three forms. Okay. So mainly there are three forms of DNA are there. Okay. Let's see one by one. So this is the B form of DNA, A form of DNA, and Z form of DNA. If you want to draw the diagram, they will look like this only. This is the B form DNA, this is the A form DNA, this is the Z form DNA. Now let's see what are the differences. See, all of these B and A are right-handed helix. Now means what right-handed helix? Suppose you are entering a house, and here it is a door. Okay. And in front of this door, there are staircases. Okay. Suppose those staircases start from your right side. Okay, like this. Okay. Suppose from here they have started. Okay. So this should be what? A right-handed DNA helix is there. So same case with this B and A. These all are right-handed DNA helix. Okay. Means that would be a turn of their entire helix would be. Now this Z DNA is a left-handed DNA. Now what that left-handed DNA means? Suppose this is that door, and the stairs start from which side? This side, your left side. Now in that way, when the stairs are left side, this entire helical turn would be from left side. So that's why they are called as left-handed. When that helix of that entire DNA is starting from the right side, it's called as what? The direction it is from for the right side, then it is called as right-handed DNA. So these are the two differences in between this B, A, and Z. Then these are the base pair per turn means what? Base pair per turns means what? See, suppose this is this DNA helix, right? Suppose this is the DNA helix. Okay. There is a fixed dimension this Watson and Crick has given that this entire helix will have a 34 angstrom as its entire length. Okay. In between there would be almost around how much? 10 base pair would be present. Okay. 10 base pairs are present like this guanine, thymine, or this guanine, cytosine, cytosine, guanine. Such 10 base pairs will be present on an average. Okay. So and the difference in or the distance in between those base pairs is what? 3.4 angstrom. Okay. And all of this data Watson and Crick has found from this DNA X-ray diffraction model. Okay. X-ray diffraction technique. So that would be the case. So in the case of this B and A and Z different form, these are their best pairs per turn. In somewhere it is 10.5, 11, or 12. Now this, let's see what is helix diameter. Helix diameter means what? This is the helix. Now what is this diameter of this entire helix is? That length would be a diameter. So that is this 20 angstrom sometime, 26 angstrom, or 18 angstrom in some cases. Now let's see what are these differences in between this major and minor group in case of this. Okay. Now what is the major and minor group? See, this would be a major group. Okay. Which one? A group which is having a much larger depth. Okay. This would be a major group. Okay. This entire. And this would be a minor group because the depth of this group is small. Same case with here. This would be a minor group and this would be a major group. Same case here. This is the minor. This is the major. Now there are certain differences among them. Now let's see what are. In case of B form of DNA, it is wide and deep. Okay. In case of this B form of DNA, this major group is wide, means this distance is larger. Okay. This is wide and deep. So this distance will also be what? This will also be longer as compared to this. This length will be also be a longer. In case of A, it is a narrow and deep. Okay, you can see in case of this A, this entire width is what? Narrow as compared to, suppose this is 4, this would be like 2 centimeter only. And the next thing is what? It is deep. The distance might be same. See, whatever this deep depth of this entire DNA strand is there, suppose this is 1 centimeter, this would also be a 1 centimeter. So that just to understand, both are deep only. Now let's see what is the case of the flat. You can see in case of this ZDNA, this even this major group, there is no much depth is there. It is kind of flat group is there. Okay. Now let's see what is the minor groove is there. The minor groove is narrow and deep and wide and shallow. Okay. Mostly we are concerned with this B, A and Z. You can see in the case of this B it is narrow and in the case of A it is wide. Okay. So you just have to remember that entire chart. So let's see now these are the main conformation of the main structures of DNA. Now we are going to see certain other structures of DNA and the notes on it which are you are very less likely to find these notes anywhere else. So just have a close attention. So let's see the first alternative DNA structure, that is what? The cruciform DNA. Okay. The cruciform DNA is like this only. It is what? A cross-shaped DNA. You can see this cruciform DNA has formed a stem. Okay. It has also formed a branch point, mean this would be a branch point, this would be a stem, and there would be a loop, means this head-like part would be a what? A loop. And among into this there would be a complementary base pairing would be there. Okay, because these are two strands of DNA, so there would be a complementary base pairing would be present. Okay, so this is what a cruciform of DNA is. Okay, and this is found in where in prokaryotes also and eukaryotes also. And what are the basic functions they carry out? Transcription of DNA, double strand repair, and even the DNA translocation and recombination. Okay. Now in this only there is a formation of holiday junctions are there. Holiday junctions are means what? Suppose these two strands of DNA. What will happen is that a part of DNA make a crossover into it. You can see this strand and this strand has now been crossed over. So such crossing over is called as what? Holiday junction. Now let's see the other form, that is DNA triplex or HDNA. DNA triplex is nothing but what? See, if there is an already formed DNA strand is there. Okay. What can happen is that there can be an additional strand can come, which will form an additional pairing or additional bonding with them, with this already formed DNA strand. So in this way now there are 1, 2 and 3 strands of DNA are there. Okay. You can see here. And what will the case mostly if one of them is 5 dash to 3 dash direction, obviously another should be 5 dash to 3 dash direction also. So whatever this complementary DNA strand would be there, it can be either parallel DNA helix also. Here you can see two are like this 5 dash to 5 dash. This adjacent parallel strands. This would be also 3 dash, and this will be also 5 dash. So in this way, this is a parallel strand. The third strand of DNA is what? A parallel strand. But what can be the other skin, that this strand can also present at an anti-parallel, means this can be a 5 dash end of this DNA and this can be a 3 dash end of DNA. Okay. Next thing is what? Bent DNA. Bent DNA is means what? See, this is the model of the DNA. What may happen because of the damage or any physical activity or physical force is that this strand of DNA can bend like this. Okay. Now there can be multiple reasons by bending this, like this ion radiation can be there, chemical reagents can be there, and wherever they will bend, they will call as what? A junction model or junction point. Then there is the next structure of DNA, that is 4-stranded DNA. The 4-stranded DNA is having what? You can see here. There are two types. Either there is a parallel one and anti-parallel one. Most of them occurs in the this guanine rich DNA. Okay, you can see in this parallel DNA quartet, all of them have arranged parallelly to each other. Okay, so that is why they are called as parallel. In the anti-parallel, what will happen? You can see this 5 dash 3 dash. You can see in this here, the all of the ends, that is the 5 dash and 3 dash ends of this DNA are not aligned with each other. They are kind of opposite to each other. So this is the 4 standard DNA model is there. Now we have seen these lots of structures among this DNA. Now we will see some of this RNA. So the thing with RNA, this will also have a pentose sugar attached to it, but only the I have already explained to you, only the pyrimidine base, that is the thymine, will be replaced by uracil. It will also be a single stranded DNA. It will be not a double stranded DNA as that of DNA. And as this is a single stranded DNA, it does not follow the Chargaff's rule. Okay. So this is the first step, that is the mRNA, and this is the prokaryotic mRNA, and this is the eukaryotic mRNA. Okay. Now, what is the prokaryotic and eukaryotic mRNA? The difference is, you can see that there is a difference in between. Suppose there is a shine Dalgarno promoter sequence in case of this prokaryotes. Okay, there will be another promoter sequence in case of this eukaryotic mRNA will be there. So you can see there are 5 dash cap is there on this eukaryotic. Poly A tail is there in case of this eukaryotic mRNA. So there are no such poly A tail or the cap like structures are there on the prokaryotic DNA. Okay. Obviously, both of them are bound to have the start codon and stop codon. This will also have a start codon and the stop codon. Okay. So these are the structures of mRNA in case of prokaryotes and eukaryotes. Now we will see the structures of tRNAs. Okay. See, tRNA, this is the one of the basic structure of the tRNA. You will have a 3 dash end with this ACC nucleotides and a 5 dash end. There will be a 4 different arms, will be five we can say, a different arms are there. The acceptor arm, why this is called as acceptor arm? Because on this arm, this tRNA or this amino acid will get attached. Okay, amino acid. On this, this is the T psi arm, T psi C arm. Then this is the variable arm. Then this is the anticodon arm. The main function of this anticodon arm is what? To attach with this mRNA. Suppose this is the mRNA. This anticodon arm will recognize the nucleotides on this mRNA, and it will form an anticodon codon attachment, and it will get attached with this mRNA. Then this is the DR. Okay. And all of these arms will have a complementary base pairing amongst each other. Right, this one. So this is the structure of what tRNA. And the last part, that is the ribosomal RNA. As we know, there are different types of ribosomes are present in the prokaryotes and eukaryotes. In the eukaryotes, it is 80S ribosomal units are there. They are having the 60S and 40S subunits. In the case of prokaryotes, they are having 70S and 70S ribosomes. They are just having the subunits 50S and 30S. And what is mean by this S? This S is nothing but the Svedberg units. Now, what is Svedberg units? See, these individual ribosomes are there. They will have a kind of their own sedimentation rate. So S just denote the sedimentation rate. So if this is the combined 80S subunit and this is the 60S subunit and this is the 40S unit, means what would the case at that? The sedimentation rate of this 60S subunit is this 60S. The sedimentation rate of this is 40S. And when you combine this 60S and 40S, whatever the units you are going to be formed, that is called as ATS. And why it is ATS? Because the sedimentation rate of this combined complex is ATS. S is what? Svedberg unit. It is the sedimentation value unit. So this was the things with this nucleic acid topology. If you have any query, you can ask. Otherwise, we will stop our session here. There are any queries? Are there? Are there any queries? And you will file whatever we have covered on this Telegram channel only, or you can just join my UPSC Zoology channel group or Telegram channel group, and also you can log into my website, that is shivaschool.co.in. On this website, you will find the free course of YouTube. Okay, in the YouTube free course, you will find all of these lectures arranged in one by one. Okay, so you can join that free YouTube course freely and can have this all of these lectures at one point. So the Rachna is saying, sir, will you take these live sessions next year also? for those who are preparing for 2022, or I will study these live now only. If it would not be available next year. No, such live sessions will be there in next year also. I will conduct it every time. Okay. But the thing is that, Rachana, as I'm already telling you that this would be a kind of shorter stuff. Introductory lectures will be there. Okay. If you want a more complete and thorough notes and the lectures also, you will have to join paid course. Okay. This is for just who cannot even afford even that 600 or 700 rupees per chapter cost also. For them, this is the free YouTube course. At least you will have knowledge on every point. Okay, so it would be not like that you will not getting a single point on any point. You will have at least 30% knowledge on every point. Okay, so this would be conducted next year also, no problem. Okay, sir, why you not upload notes on Telegram? Which one? Which notes? The notes are there for the paid group, na? Okay, the Mohammed is asking that question, why I have not uploaded the paid group. Okay, and if you are asking for this PowerPoints of these, then I can upload these. Okay, no problem for that. Okay, I will upload this PowerPoint. See, this thing is that there are lots of things happening. Because of this only, I was not able to conduct these live lectures because of paid batch. So if you want these PowerPoints, I will upload it. No problem. Okay. So I think I'm not. Whether any queries are there? These paid courses will be live. Yes, these paid courses will be live only because see this we have conducted first cell biology course from 6th November to 22 November. Yesterday we have finished this course only. There were almost 55 students who were joined this course, and all of these are live courses, and you will also get the recording of the same lecture. So even if you join the right now only, you will get the recording of the lectures, which are more than 30 hours of lectures are there on the cell biology, and along with that for 600 you will also get a printed notes on cell biology. Okay. Yeah, Rajan Soni is saying good morning. Yes, good morning. Yeah, these topics. Yes, I will upload these shorter PDF notes also in the group. You can access the short PDF notes in the group. Okay, Mohammed. Okay, I think there are no more queries now. So we will stop this session here, and we will meet tomorrow same time, 11:30, with the next chapter. And it would be like mostly it will be the mitotic spindle and chromosome type and chromosome movement. Okay. So that was one of the just topic which have been remained. So let's see, even if there are some other points are there which are remain like protein transport and folding, we will conduct that also. Okay. So we'll stop here. No more queries. Okay. So have a great day and bye bye. We will meet tomorrow 11:30. See, Rachana is saying timing for paid live session. See, Rachana, you can join my Telegram channel. There you will get a lot of information on this entire course. I have also made a video on this also and uploaded on my channel, so you can watch it. The timing of the paid batch, the new paid batch for the now we are going to start the physiology paid batch from the 1st December. We have already completed the cell biology. So the timing for that physiology would be 9 a.m. to 11 a.m. Okay, in that you will attend this live session, and after that you will get the recording. Okay, and it is 9 to 11 because you can just complete the most of the part of optional like for up to the 11 only, and then you can study for your current affairs and the GS. Okay, so I hope I have answered your question, Rachana. Okay, and if you have not joined my Telegram channel, search on the Telegram Zoology UPSC IFS. There you will get a link for the channel also. It is having 7,500 members in it, and in that only there will be a group for a discussion group also, so you can join a discussion group also. Otherwise, if you have any query, you can directly contact me. My contact number is shared in the description also. Okay, so you can contact me directly for joining it. The cost of this new physiology module is 750. Okay, and if you are also purchasing the cell biology at the same time, you will get a 50 rupees discount, so that will be 700. Okay, so right now we will stop. We will see. We will meet tomorrow 11:30 a.m. So have a great day. Bye bye.